All right, now moving on to the basic functions that happen in a cell. And again, a lot of this is going to be review, but this is information you're going to need for this class, so I want to make sure we're all starting on the same page. Thinking back to this simple diagram of a cell, there are some elements that we'll recognize. So we've got the plasma membrane that's made out of a phospholipid bilayer, separates the inside of the cell from the outside, is very important for moving nutrients inside the cell, getting signaling across the cell. So that's thinking about our plasma membrane. Some of the other parts of the cell, we've got our nucleus, which is where the cell's DNA is stored, so your chromosomes, that's where they're stored. That's where DNA is transcribed into RNA for making proteins. That's also where DNA can be replicated into additional copies of DNA if this cell is about to divide. We've got some other things like the rough endoplasmic reticulum. You see that this membrane fuses with the nuclear envelope. And this is important for making lipids. This is where new lipids are made. Also, these rough spots, that's where ribosomes, which remember ribosomes produce proteins. So ribosomes sit there at this membrane, and that's where transmembrane proteins are made. So anytime you have a protein whose final job and role is to be a membrane protein, it's going to be made in the rough ER. The rough ER then feeds into the Golgi over here, which helps sort and package molecules and deliver them where they need to go in the cell. So for example, was that a molecule that needs to be secreted outside of the cell? The Golgi will make sure it gets there. Is that a molecule that maybe needs to be trafficked back to the nuclear envelope? The Golgi will make sure it gets there. So we talked about the rough ER, the Golgi. We also have the mitochondrium. As single as a mitochondrium, multiple are mitochondria. So the mitochondria is responsible for energy production in the cell. We're going to talk more about mitochondria later. We have a lysosome, which is a membrane-bound vesicle inside of the cell that contains enzymes inside of it that break things down. And it's got an acidic pH inside of there. So whenever the cell needs to break something down, that could be breaking down some food that it ingested into smaller components. That could be recycling its own components inside of the cell. So if the cell doesn't need a certain protein anymore, it can break it back down into amino acids and recycle it. All that happens inside of these lysosomes. And this is what I think is absolutely incredible. We've seen so many of these diagrams of cells that are just cartoons. Somebody drew them, but they drew them like that because they saw that with electron microscopy images and now fluorescence microscopy images. So all of these sort of cartoons come from real images of cells that were taken. Again, you've learned about these parts of the cell, even in high school biology, so just to make sure you got it, take a couple seconds, jot down a few words about the function of each of these. I'll have you do this in class, but for now I'm just going to move on. All right, and then a reminder of some basic cell functions. One of them is DNA expression and protein production. So we talked about the DNA, which is stored inside the nucleus of the cell. That DNA is then transcribed into RNA in the nucleus. So here's our double-stranded DNA. Those strands are separated. An RNA polymerase enzyme comes in and makes this new strand of RNA that you see here in this teal color. So adding it nucleotide by nucleotide, we have this new RNA strand that's made. That mRNA, so M is for messenger RNA, is then trafficked out to the cytoplasm of the cell, so outside of the nucleus, but still inside the cell. And then ribosomes will translate it. So now it reads this code where 3 nucleotides in the RNA code for 1 single amino acid. So this is translation, reading the RNA code and translating it into amino acids. So these amino acids are brought in one by one and then covalently bound to the amino acid in the chain before it. So then we get what's called a polypeptide chain, and this is what makes up the protein. So this amino acid sequence will then fold into the protein. So we can see how the genetics, a person's genes, will directly impact what amino acids end up in their proteins and how that protein is going to function. So this leads back into enzymes. Again, remember that enzymes are proteins that catalyze reactions or builders and breakers in the cell. So here's a more specific information about an enzyme where we have our cartoon enzyme here. So this is a protein and it has an active site. So then some substrate, here's another example of glucose and fructose bound together. That's the substrate for the enzyme. So that's what the enzyme is going to act on. That will bind the enzyme's active site in a very specific way, such that most of the time only this substrate can bind this enzyme. Together, they form an enzyme-substrate complex, and then there's some chemical reaction that occurs, in this case adding a water molecule, and then the glucose and fructose are released as individual molecules. So again, we've done some chemistry here in the active site of the enzyme where we've broken the covalent bond that was holding glucose and fructose together as a single molecule, and now they're separate molecules. So these are the products of this enzyme, and then the enzyme is released and can be reused to catalyze another of these same reactions. Here are some examples of digestive enzymes. So anytime a biochemical word ends in ase, that's a pretty big clue that it's going to be an enzyme. So if one example is amylase, which is in our saliva, which breaks down starch into maltose and eventually glucose. So we also have gastric juices, so protease, which breaks down proteins. We've got pancreatic juices, again, all these enzymes. You can tell they're enzymes because they end in ase. Not that I need you to memorize these, but just to give you some examples for now to make it a little more relevant. And we're going to spend a big chunk of this class talking about enzyme inhibition. Why is that important? Because the body has several natural mechanisms for inhibiting enzymes, and enzyme inhibition is a major consideration for pharmaceutical drugs. So here's one example. Here is the enzyme that is targeted by penicillin. So penicillin, which is this molecule here, is an inhibitor for the enzyme that synthesizes the bacterial peptidoglycan layer that stabilizes the bacteria cell membrane. So here's a bacterial cell, gram-negative or gram-positive. Let's use gram-positive as our example here. There's the cell membrane, the inner cell membrane, which is a phospholipid bilayer, but then outside of that, gram-positive bacteria have this peptidoglycan layer, so peptide, glycan, sugar, so protein sugar layer that helps protect the cell from other things that might want to come destroy it. It also gives the cell extra structure and shape. And this peptidoglycan layer is important for this gram-positive bacterial cell to be able to survive. So penicillin inhibits the enzyme that makes this peptidoglycan layer. So what happens is when penicillin is added, that enzyme is inactive, this peptidoglycan layer can't be formed, and the cell structure is unstable, and the cells burst and they die. So that's why penicillin is such a potent antibacterial medication, because it's an enzyme inhibitor. So we're going to talk about, like I said, spend a lot of time talking about enzyme inhibitors, and this is why it's important from a medical perspective. All right, another key cell function that's going to come up in this class is membranes and cell communication. So here's another diagram of a cell, and we've got the plasma membrane, again, surrounding the cell. Plasma membrane, cell membrane, same thing. So this membrane is a semi-permeable barrier. What that means is that only specific molecules are able to cross the cell membrane, which is important because that's basically how the cell defines itself from its surroundings. Things inside the cell are obviously part of the cell, and things outside are not a part of the cell and can't get in unless they're one of those specific molecules that can cross that semi-permeable barrier. So that's one important function of the cell membrane. Another is all about signaling. So we talked about those transmembrane proteins that can receive signals from outside of the cell, and most of the time those signaling molecules are not passed through the cell membrane. They cannot cross that semi-permeable barrier. So instead there's a protein here that acts as a receptor, and then that protein will pass the message. So not the signaling molecule itself, but the message, and we'll talk about this in more detail later, what this really means. But it passes that message to some intracellular or inside the cell molecule that will relay the signal to cause the downstream change that that cell needs to do to respond to that signal. So that's called a signal transduction pathway, where that message gets passed from molecule to molecule. And then the end result is usually changing a protein's function. So inactivating a certain protein or activating a certain protein, or regulating gene expression. So turning on production of a certain protein or turning it off based on whether the DNA for that gene is transcribed into RNA or whether it is not. Just to make this a little more real, here's an example of the pathway for adrenaline signaling. So adrenaline interacts with this adrenergic receptor, so that's a transmembrane protein. This is called a G protein coupled receptor. We'll talk a lot more about that later. It interacts with the G protein, which then passes the signal to adenylyl cyclase, which increases the production of cyclic AMP, which is a very important signaling molecule, which interacts with pKa, another important signaling molecule, which goes into the nucleus of the cell and then phosphorylates this protein that regulates transcription, and then that activates the expression of this target gene. So then this DNA is transcribed into RNA, which will go on to make that protein. I don't care so much that you know all the parts of adrenaline signaling right now. I just wanted to give you a real example of membrane-mediated cell signaling, so it gives a little more context of what we're going to be talking about and why it's important. So practice question, you can work through this on your own, make sure you're following along. We'll also discuss it in class. So consider a hypothetical enzyme oxidase, just made up oxidase, that makes reactive oxygen species in response to inflammation signals from nearby cells. Propose 2 biochemical methods you could use to reduce how much oxidase is active in these cells. So think about the concepts we just talked about and think about how you could use those to reduce how much oxidase is active in these cells. And then finally, we're going to finish this up by talking about mitochondria. Mitochondria are going to be very, very important when we get to energy production. So I want to make sure you have that foundation of knowing what they are. So mitochondria have a really, really interesting origin story. They likely originated from a symbiotic relationship with a bacteria. What do I mean by that? So there was likely some bacteria that was a really good energy producer, and a early eukaryotic cell engulfed that bacteria and then kept it. So then that bacteria permanently lived inside the eukaryotic cell and made energy for the cell. And then the mitochondria, so that bacteria, divides on its own. And then when the eukaryotic cell divides, it just has some mitochondria in it, sort of randomly when the cell splits into 2 cells. So the idea is that mitochondria look a lot like bacteria when we look at what they're made out of. And mitochondria also have their own DNA. So the idea is that we think this used to be an independent, functional bacterial organism that then was brought into a eukaryotic cell and has stayed with it ever since. So a symbiotic relationship is where two things are living symbiotically, meaning that they're helping each other out. So the benefit to the mitochondria is it gets all the resources that the eukaryotic cell has. And then the benefit to the eukaryotic cell is it gets all the energy that's produced by the mitochondria. So mitochondria, like I said, they have their own DNA. We can't see it in this diagram, but if we did, we would see DNA here. It's not stored in a nucleus. There is no nucleus inside the mitochondria. It's just DNA that's inside of there. And that DNA is transcribed into mitochondrial proteins, but the mitochondria also use proteins that come from nuclear DNA. So imagine off somewhere else in the cell is the nucleus. Inside that are the chromosomes. That is transcribed into proteins and transcribed into mRNA, translated into proteins. Those proteins are then trafficked into the mitochondria, and some of those also help the mitochondria function. All that being said, we're going to focus on their role in making energy for the cell. Now, the way this happens in a very broad way is that inside of the cell, again, we have the cytosol or the cytoplasm, which just means the inside of the cell. We also have some mitochondria. So in the cytoplasm, glucose is broken down to pyruvate in this process of glycolysis, which we're going to talk a lot about. This makes ATP. It also makes this other molecule NADH, which is trafficked into the mitochondria. Pyruvate then gets fed into the citric acid cycle, which makes more ATP and more molecules of NADH and FADH2. These, all three, get fed into oxidative phosphorylation, which is a process that happens inside the mitochondria. This produces, again, a lot of ATP for the cell. And this whole process overall is referred to as aerobic respiration. We're going to go into this in a lot of detail later, just giving you the background of why it's important. And then finally, just some vocabulary. So you need words to be able to describe the different parts of the mitochondria. Luckily, most of them are pretty self-explanatory. So we've got the outer membranes. This is a phospholipid bilayer called the outer membrane. We also have the inner membrane. So you'll notice there's two phospholipid bilayers inside of the mitochondria, the outer and the inner. And the inner has these curvy structures here. So lots of membrane surface area for the inner membrane. We have the intermembrane space, so in between the outer and inner membranes, that's called the intermembrane space. And then on the inside of the inner membrane, that's called the matrix. And then these little folds here, sometimes if we get a fold that's completely enveloped, that's called a cristae. So just giving you some vocab for mitochondria. Again, those are some basic cell functions that are going to come up throughout this class. A lot of it's probably review, but just bringing that knowledge back to the forefront of your brain.